Okay, thank you. So some of you have been listening to me like 1 year ago or 2 years ago, I don't remember, in Bertinoro, so where I talked a little bit about basic phylogenetics. So in a sense, this is a little bit of a continuation of that talk, but I'm going to go into tumor, okay? So as the ones that were there in Contra, you know, I'm an evolutionary biologist, I'm a phylogeneticist that recently moved to cancer. So what I'm trying to do is to move the tools we use in evolutionary biology, which are old and well understood, into cancer, right? So that's going to be the second part of the talk I gave you in Italy, like last summer, I don't remember. The first chapter is a little bit of remind you what is cancer evolution and genomics, the stage. You already have seen this slide. So this is so-called Darwinian evolution of somatic evolution, a healthy cell, and one mutation or many mutations, it doesn't matter, that becomes tumoral. And then you have a blue clone, and that blue clone evolves, okay? The evolution of species, individuals, genes, alleles, haplotypes, whatever you want to call it, in evolutionary biology, we normally draw it as a tree, okay? So a phylogenetic tree that in this case, if we knew this phylogenetic tree, right, we might learn a lot of things about what happened in this particular patient, right? About surviving a selective pressure, maybe from the immune system, about extinction of clones, about some clone that gets big and travels to a different organ, another one that is small that travels, therapy, recurrence. So a phylogenetic tree is going to describe the evolution of this tumor, and the evolution of the tumor has to do with the disease itself. So somatic and germline evolution is evolution so far. So most of the research in evolution has been on germline evolution, okay? The process of change across generations, okay? So we can define evolution as genetic change across generations. That's one way of defining evolution, okay? So we have a split, no, from the mouse long time ago, a couple of million years ago, human and chimp. Then we have human populations. This is us in this room. We are here. And maybe some person at some point. Well, everybody there is somatic evolution, in all of us, within ourselves. In this room, during the 40 minutes of these talks, we are going to accumulate 100,000 mutations, okay, in our cells, more than that, okay, in every mitosis. And in some cases, if one of these mutations hits the wrong site, right, we get a driver mutation, and then we get expansion and control cell proliferation that we call that cancer, right? So all this process can be seen as just one process, but there are fundamental features, okay? In germline evolution, there is a recombination because transmission across generations, not in all species, but in many species, is through meiosis, to the sex gametes, right, which makes the genome, different parts of the genome to be independent from each other. And that has like very profound consequences on the evolutionary process. Very profound, conceptually, technically, methodologically, statistically, at all kinds of levels. That's a very big difference. Then, of course, the time scale is very distinct. We're talking about thousands, millions of years, right? Millions and many generations, so we have a lot of mutations. Somatic evolution, the time scale is slower, but it's the same process. It's mutation, accumulation, right? Drift, selection, migration is the same thing, but it's across years, right? Lifetime of a person. And of course, the process that is most important is mitosis, cell division. There is no recombination of the genes, so the whole genome is linked. So there is just one locus, in fact, okay? The whole genome is one locus. This has some implications. So there are challenges to study somatic evolution compared to study germline evolution. So I already mentioned this because there is a short evolutionary time, there is lack of variation. We don't have as many mutations as we could have if we study with the same techniques, right? Different species, right? The number of changes between different species is much larger than between cells of a tumor in general. So therefore, to apply sophisticated statistical techniques that need information might be more difficult in this case. I already mentioned this. The implication of a single locus from a statistical point of view is that we don't have replicates of the evolutionary process for a single tumor, right? when we study germline evolution, different loci can be independent replicates of the evolutionary processes. So therefore, we could have the same expectation for the point estimates, but the variance is much larger in somatic evolution of our estimates. It's expected to be much larger. Then selection is assumed to be extremely important in cancer, right? Well, in germline evolution, there's been a debate in the 70s. The neutral selectionist debate happened 50 years ago in germline evolution, and it's solved in a sense that most of the processes are neutral and there are some selective processes, I would say, in general. This is not completely true, but what is true is that most evolutionary models for germline are neutral. Okay? The assumptions assume neutrality and also independence of loci. Okay? Structural variants are, pardon me, not that important in germline evolution. Okay? We don't get that many structural variations as in tumors. I think that's fair to say. So most models in germline evolution are for non-structural variants, for SMBs, in fact. Okay? So therefore, I would say that we have poor evolutionary models for structural variants. Doesn't matter for germline or somatic. We have poor models for structural variants. So therefore, we have poor models for structural variants in cancer. And then the way we sample is totally different. The way we sample tumors than the way we sample frogs or Drosophila flies in a population. And this has implications. Okay. Well, the outcome of this evolutionary process of that tree is that population diversity. That's nothing unexpected, right? So in any population there is diversity. In the long term, it might be changed in different times, but in a tumor, we have differences between tumors across patients, but I'm going to talk about what happened in a single tumor, okay? So I'm talking about evolution in a single tumor, and that's actually how it happens, in fact. So we have different regions that may have different clones, different mutations, and of course, normally we get a single sample from here, okay? And that's might be a bad representation of this heterogeneity. So we have to deal with this, right? Another strategies that have been appeared in recent years, not in the clinical setting, but in the academic setting, is multi-regional sampling, okay? That logically, we get a better picture of the whole tumor if we get different spatial samples. It's very obvious, right? And of course, the standard way of sampling tumor is bulk. So we get DNA from millions of cells and we mix all this DNA, and that's the standard data that we have in the databases, is bulk sequencing, right? That is the equivalent of going to the field, getting 100 frogs and put them in a single glass and get DNA from the 100 frogs. So we don't have individual identities, okay? And individuals is the unit of evolution, so, or one of the units level, okay? So in the 10 years ago, single-cell sequencing appeared. It's not still very common for DNA. It is very common for RNA, okay? But to study genotype evolution, the evolution of the genomes, we need DNA data, okay, in general. And this is the ultimate level of resolution. Of course, we can get also single-cell data or single-molecule data from the blood, okay? So we are in our lab using all these techniques, in fact, okay? Now, what are the implications of sampling bulk? Is that, well, nowadays I would say that the bioinformatics, given enough coverage, we know more or less how to do. So we get DNA from these million cells, we put it in the machine, and then we do the bioinformatic analysis, and more or less we know how to identify variants. No? If you have 100x, probably your variants are going to be quite robust in general. Okay? But when you isolate a single cell, you just have 2 molecules of DNA, right? 2 double helix. So we need to amplify the DNA, right? So in this process, when we amplify the DNA from a single cell to get a similar amount of DNA, because we need a minimum input to the machines, right? We need to amplify. In this step, there are multiple biases that occur that are going to complicate the interpretation of the data. So the data is much more fragmented. Specifically, when we have single cell, what we have is that there is coverage variation. Okay, this is biochemistry and physics of just dealing with 2 molecules there, right? We need to amplify using enzymes 2 molecules, okay? So basically you get, while in a population, this is bulk sequencing, the coverage more or less is uniform, might follow a Poisson, might be more or less over dispersed if you want, but For single cells, there is a lot of dispersion of the coverage. So this cell has a lot of coverage for this part, few here, and so on. And the problem is that the next cell, the coverage is going to be again different, okay? To some extent, okay? I'll tell you later, or not, because I think I don't have that slide. But coverage changes across different cells, and that's problematic for interpretation of the data, okay? So you have this phenomena also, you have a coverage variation, then you miss alleles because either you hit this molecule in the first time or you start amplifying and then you miss one of the alleles and you don't see it in your data, right? So you can have an heterozygote, you just don't amplify the B allele and you think you have an homozygote. So you are missing the B mutation, right? The false negative. You can miss one allele or both, right? That's usually called allelic dropout, and this is called locus dropout. And moreover, we know that we have a more or less constant sequencing error rate, right, in our grids, and we know more or less how to deal with this. But amplification error might be very different, okay, because there are amplification errors because we use polymerases to amplify the DNA, that they have like an error rate. And if this error happens at the very beginning of the amplification, this error is going to spread in your pop reads, and then most of your reads are going to have this error. So therefore, you're going to be very happy that you are very confident on your variant, but it's totally wrong, okay? So amplification error is much more problematic than sequencing error, okay? So you get this type of things, false positives, okay? And also false negatives. You get false negatives from amplification error, right? If this mutates and it looks like homozygote when in fact it's a true heterozygote. Okay, so these are different things. Okay, so that's basically the type of biases that we need to deal with single cell. So I just put this slide because Holger was this morning here and then I remind me that. So we have this paper now in second round review, it's in bioRxiv, so this is like a wet lab experiments we've been doing the last 2 years to check which is the best protocols to avoid these biases, okay? And different kits have different properties. Some are better for allelic dropout, some are better for amplification error, therefore some are better for copy numbers, some are better for single nucleotide variation. There are different properties. I'm not going to talk about this. Some are better, some are more random for the coverage, some are less random. So it's all written here, so you can go and it's open and you can see the technical thing, right? The point is that there are some kits in the market that, we should be used depending on the question we want to address. Okay, and the answer is here. Well, how is going to look the data for single cells? The data for bulk, it will be something like this. Normally in cancer, you have a more or less complete matrix where you only see 2 alleles for a single SNV because given the mutation rate and the time scale, normally there is no enough time to have 2 mutations in the same site as an expectation, okay? So most sites are biallelic and so on. But when you have single cell, things change, okay? Because you're gonna miss data because you have allelic dropout, and then you're gonna start to see in some portion of the data, depending on the amplification error and also the mutation rate in that site. This is like an overall, okay, view. You might have sites with 3 alleles, and then you might have like multiple genotypes and so on. It's much more noisy. It's more sparse, okay? That's the data we have to build phylogenetic trees from single cells, okay, in cancer. Now, second, trees. What is a phylogeny? A phylogeny is the evolutionary history of what we call taxa, okay? Taxa can be traditionally a species, right? Human, chimp, mouse, the history of this species, but can be alleles, can be genes, so we use it in different frameworks, can be individuals, can be populations in some cases. The phylogeny is the history, and the tree is the depiction of that history, okay? We have use of the word phylogeny. Phylogeny is what it happened, and the tree is the depiction of that, okay? It's the graphical representation of a phylogenetic tree. But, importantly, not everything that looks like a tree is a phylogeny. Phylogeny is about evolution. You can build a tree with the plate of cars. You can build a tree with, the colors of the church gears. You can do clustering of many things. You can build clustering trees of many things. These are not phylogenies. Phylogeny is about evolution, about ancestry, about sharing variation because of ancestry. So this is an example where this phylogeny of across patients it's not a phylogeny. It's a tree, but it's not a phylogeny. I don't know why it's here, right? So there are multiple examples. Next, I'm going to show you how sample trees, very common in cancer and in many publications, are not phylogenies neither. Okay? So at least we should use the proper name. Phylogeny is not similarity. If phylogeny was similarity, I wouldn't be a phylogeneticist because it would be trivial to build phylogenies. Okay? So phylogeny can also be thought as the art of disentangled homoplasy, convergence, disentangling the fact that two things may look alike but be unrelated. That's the art of phylogenetic inference. Okay? So imagine that this is the true evolution of these three species, if you want. We don't have often, in evolution, mutations accumulating at the same rate for multiple reasons. Might be generation time, might be the mutation rate itself, but it's often the generation time and other properties of organisms. So in this case, A and B share an ancestor, but B evolve very fast, like birds within reptiles. So birds are reptiles. The question is they have feathers because they evolve very fast, different features, okay? So in this case, this is the true phylogeny. If you just think of similarity, C is much more similar to A is much more similar to C than to B, just because of the number of changes that they accumulated, right? So you are going to always infer something like this, okay? So phylogenetic reconstruction, and we know this for many years, is not about similarity, okay? It's about finding common changes in the ancestors. Otherwise, you are not doing phylogenetic. It might be fine. You don't have to do phylogenetics. You can do whatever you want, but at least coin it in the proper way, okay? Phylogenetic is about evolution, about search changes. It's not about similarity. Obviously, there is a correlation between similarity and phylogeny, obviously, but it's not an equality. It's just a correlation that might be stronger or weaker, depending on the situation, okay? What type of trees do we have in cancer? So we have true trees, that they exist, and then we have inferred trees that we get from the data. So the true entities that exist in cancer are cell trees, because cells are related, no? Through mitosis, cells divide in two, and again, and again, and again, and that history is independent of mutations, okay? The history, the genealogy exists, even if the cells are identical. Even if there are no mutations, the history exists, okay? So don't get confused. One thing is history and other thing is mutation. But we use mutations to decipher the history. That's true. But those are two different things, right? And then we have the history of genes, alleles, clones, haplotypes, whatever we want to call that sequence of mutations, okay? We have different names for more or less the same thing. Because in cancer, there is no recombination in principle, the cell tree is the same as the gene tree, in this case. In other cases, it's not like this. But in this case, it is like this, okay? Now, we infer trees from multi-regional trees, okay, like this paper, clone trees, like this here, single cell trees, like different naming, and mutation trees, okay? But these are products of our inference, and the only true trees, true phylogenetic trees are these, okay? You can use these type of things for different purposes, but be aware, you have these things that you can build, but truth only belongs to these two type of entities, okay? So that's important to distinguish. Multi-regional sample trees are probably the most common representation. So this is a seminal paper by Marco Gerlinger, where you have different regions, and then you can see that you can disentangle convergent mutations and parallel mutations in the same gene and so on. But we need to be aware of this. All these trees are built from bulk sequencing. So when you get a sample from a given time point, you're going to get a mixture of clones in your sample, okay? So the way I look at this is like this, okay? So a bulk sample is what we call pool sequencing. What I told you before, I go to the field, I get 20 frogs. We don't do it with frogs, but we do it with Drosophila. To do things cheaper, it's just because of money. You can get 20 Drosophila that you think that are from the same family, that they are related, and then you put them in the same tube and then use that as a whole thing, okay? That's called pool sequencing. So bulk cancer sampling, tissue sampling is bulk sequencing, okay? But the difference is that when I go to the field, because, so I work here, this is Cape Verde, so we go there, dive and get snails, okay? I can see their features. I can recognize the populations. I can recognize morphological features. I can recognize geographical accidents, beaches, currents, so I can have a prior information of what a population is. And a population is a set of related individuals, okay? But in cancer, I have no clue. I mean, do you really think that the pathologist, when it cuts pieces of the tumor, is sampling populations? Not at all. It's sampling cells, but not populations. In germline evolution, if I can go to a forest and then to a different forest, and there is nothing between the forests, I can recognize things, okay? Only when you sample metastasis, you might be more or less sure that you are sampling a different populations in the evolutionary concept of population, okay? So basically, you are sampling a bunch of cells. And this has consequences. So if you want to read this paper, where we entitled this multi-regional trees are not phylogenies, although these are the most typical representation of trees in cancer, we have some cartoons here to illustrate, because this is just very simple. Once you get, this is the true history of these clones, but when you sample, you can get a mixture of clones from each sample 1, 2, and 3. And when you can get a mixture of clones, and you apply a phylogenetic method over the populations, you're going to get apparent homoplaces, changes that you're going to think that happen twice, but in fact it's because you got mixed populations. It's an artifact of the data. It looks like there are 2 parallel mutations when there was only 1. The question is that the clone that carries that mutation is in 2 places. And if these 2 places overall seem to be more unrelated than other places, you're going to infer 2 independent mutations when only 1 happened, okay? That's what we call homoplasy. So not only that, the implications of using sample trees is that you get sometimes, I'm going to stop, you have the paper, so I'm not going to stop that much. The tree that you infer might tell you a different branching order, and that might be important for the metastasis. It might confuse you, and you might get the impression that one metastasis happened earlier than other, when that's not the case, okay? Because you are mixing clones, and you don't know which clones you are mixing, okay? If you don't do clonal deconvolution, of course. And then wrong branch lengths. The length of the branches is going to be spurious. And if you want to infer things like time or mutation rates, you're going to get confused. So think about this. Do you think there is a phylogeny of New York, Calcutta, and London? Is there a phylogeny for these cities? Doesn't make sense, right? What is a phylogeny? Is closer London to New York than to Calcutta? Or to another city? No, because there are not evolutionary units. London is not an evolutionary unit. New York is not an evolutionary unit. A sample from a tumor is not an evolutionary unit. It's a mixture of cells. It might be, but it doesn't have to be an evolutionary unit. When I go to Cape Verde and go to a beach, I have a prior information about the color of the cells, the location, the behavior of the animals. So I have a prior information that might be a population, right? That has a common origin, a common ancestor. But in a tumor, there's no way of knowing this. So please avoid sample trees to make inferences. So first measures, okay. Is this real? Do we get these type of things? Yeah, doing a simple study with the TracerX, the first 100. So we just counted mutations that are shared among regions, and then you can get a lot of incompatibilities, because if one mutation is shared between region 1 and 2, and other between 2 and 3, another between 1 and 3, one of these mutations cannot be real in the sense that occurring only once, because you cannot be supporting 3 branches at the same time. Well, I can explain that further, but 25% of the mutations are apparent homoplaces, are not true homoplaces. Of the homoplaces that you see, convergent changes are apparent, are artifacts due to use sample trees. So this is a real problem, okay? So you might say, so David, what is the solution? Well, we should use evolutionary units. Where are the evolutionary units in a tumor? The clones or the cells. Therefore, one option when you have bad data is to do clonal deconvolution. You know what is clonal deconvolution? Maybe you got some workshops about it. I don't know. Clonal deconvolution is a very tough problem, right? Basically, it's getting bulk and from the allele frequencies of the data we try to say, well, I have 3 clones and this is their prevalence in this sample. Okay, it's a difficult one. It's based on some rules. It makes some assumptions about infinite sites models and other things, right? And well, they seem to work fine in some simulations, but it is a very difficult problem. So can be confident or not. I think that we still need to learn more about the behavior and the performance of these methods, right? Because in ideal situations they will work well, but in non-ideal situations we don't know how much. There are many of them. This is a list. There are many methods, and there are a few limited benchmarks in my opinion. I think we are still missing like a comprehensive third-party benchmark. Well, you will get like a clone tree in this case, and this is Florian style, so that's Florian is not here, but I put it just in case. This is his graphical style, so basically that's a phylogeny. This is a phylogeny of alleles, clones, where you have mutations, accumulations, and you might miss some of them, you get your samples, and so on. Cells, and so on, okay. And then the other type of data, apart from clones, is single cells, okay. Single cells are ideal because all the evolutionary theory methods are designed for individuals, and cells are the individuals in the tumor scenario, right? So here, this is data from nicknaming. It's difficult to get. You get biases, but it's possible, and we are producing this. And finally, mutation trees, which in some cases they can be quite complex. I'm not sure what to make of this figure. I think this is phylosub inference. I'm sometimes confused, but this type of mutation trees. So these are the type of trees. If we want to do single cell, there are several methods, right? Some of them have been developed by people in the network, like Nico and Florian, okay? And we have developed our own one, okay? Just porting methods from germline phylogenetics. So basically, the idea of our model is just to think that, well, if there is no recombination, the history of the maternal and paternal chromosomes is the same. Okay, when there is meiosis, instead, there is a segregation of chromosomes, and this will not be true again. But mitosis is an ordered process. It has to be perfect, right? You should receive chromosome 1 to 23, right? And then chromosome 1 from 23 from your mother and your partner. It has to be ordered, perfect. So everything goes like this. So the history of maternal and parental alleles is the same in the absence of recombination. So we can think of this as this, okay? The cell tree is the composite maternal and paternal history. Having this, and what I thought is that I could just apply the models we use in phylogenetics. This is a Markov model where we have like a probability. We don't need to assume anything about the mutation rate, anything about the possibility that a site changes one. We don't need to assume anything. We just have beginning states and ending states in the phylogeny and a probability of having the same state here or a different state, regardless of the number of changes or whatever, right? This is the standard way of modelizing the substitution process in phylogenetics, in germline phylogenetics. And then we can get the likelihood of a tree given the data. And then we maximize this value, and then we get the maximum likelihood phylogenetic tree. We can also move this to this. So what we did is just, instead of using a 4x4, sorry, this is ACGT. So we have like a genotype, a diploid matrix. It's the same idea, but instead of a 4x4, it's a 10x10. And we have parameters for changing from CG to AT to whatever. We could extend this and include codon models, DNDS models, and so on. And then we can also take into account uncertainty. Why? I told you before there are many biases in single cells, in single cell genomics from the genotype. So you are not sure. When you use Sanger sequencing normally, in the past, you would be quite confident that if you have an A, it's an A, because you will sequence both directions and you'll see an A and say, I have an A. But here in single cell, you might see an AT at this position, at this SMB, but you are not 100% sure, not even 99%. So we can modelize this in different ways. We can do it internally, right, to estimate the uncertainty due to alec dropout and amplification sequencing error. We don't distinguish between this and this. Okay, that's very difficult. It could be done, but we just miss it in a single parameter, and then we do the math, and then we have these equations to apply at the tip of the tree, because this is a question about the observation of the data. And the data is here, only at the tip of the tree, right? So allelic dropout is not an evolutionary process. Allelic dropout is a technical artifact, okay? Only happens at the tip of the history. Or we can use genotype likelihoods. So if you have a variant caller that gives genotype likelihoods, the data, instead of being 80, is the 10 genotype likelihoods, okay? And that improves inference a lot, as I'll show you. So sorry, because I'm going to have a few graphs. I'm going to try to, so this is, I developed, what I did with this is that, so I have the model in my mind. So I talked with some friends that have been many years optimizing phylogenetic inference with all kind of computational tricks, and they wrote a program which is RAXML, who has tens of thousands of users, I don't know, maybe 100,000 users. So it's very optimized to do these calculations. So we just put the model in. Alexei was the one that did it. And we did simulations. So we estimate the parameters. We estimate allelic dropout. We estimate amplification error. 5 minutes. We do it very well. We get very accurate maximum likelihood estimates. Infinite side models. We are this last part. If this is error, so it's better to be in the lower part. So with different genotype errors and allelic dropout. So we do, when there is no error, a simple method like parsimony is better. But when you start to have allelic dropout or amplification error, things change, right? And our method is at least best or equally best always. This is under infinite site models, so you don't allow mutations to happen twice at the same site, right? But infinite site models is an abstraction made by humans. It's not real. Evolution doesn't know about infinite site models. Evolution, mutation just happens or not, okay? It's just a question that when time is short, it is unlikely that you have two mutations at the same site, okay? But we have also, I mean, when you do it with signatures, same picture again, we get better, and parsimony gets only good when there is no error, then it goes bad, but we are better than competitors in our simulations. When we do finite site models, when complicated things, mutations can happen on top of each other, we are much better than the competitors in this case, of course, because we use finite site models. So we use a Markov model that doesn't care about multiple substitutions on the same site, or doesn't care. We are much, much, much faster than the other tools. To give you an idea, the second faster tool is CFIT. It takes 50 minutes and 16 threads, where we do in 2 minutes in 1 thread. So we are 300 times faster than CFIT. And when we use the likelihoods, and the data as a BCF with genotype likelihoods, we are these three with different flavors of our program. We get much, much better in the complex situation, which is this one. We are the best, and in the simple situation, we are also the best. But the difference grows with low coverage. With more coverage, things are more equal, but still we are the best in our case. Okay. In practice, well, sorry, I'm going to have to being slow. This is the tree for an evolutionary biologist is the beginning of the analysis, not the end. We use trees to infer things. So this is real data that we produce in our lab. We infer 21 clones. We infer the tree, maximum likelihood. So we can infer that there is only a single origin of all the metastases. So there was a single metastasis in different organs. We can infer also identify positive selection on the branches. So we have methods to identify DNDs on branches. Curiously, the branch with the highest evidence of selection is the one that leads to the metastasis. We can identify putative metastatic drivers using these techniques. We can date, so we use something called relaxed clock. This is based on inference on this. This tumor, for this person, we infer this interval, 7 years, the age of the tumor, and very early metastasis. We can infer the demography with all these methods also, that there was like a steady stage, then growth, and then a steady stage. In fact, the metastasis diverge faster than the primary tumor, which is not surprising because these cells have more mobility, right? So I'm going very fast, so you can ask me. But this is in bioRxiv also, this paper. We also use biogeographic models, which are in fact, in my opinion, much better than Machina. Why? Because if you know Machina assumes that a clone can live in one location only, because it uses a very simple parsimony reconstruction. So instead of that, we use a Markov model where the states are the ranges, the set of locations that a clone occupies. And we can infer ancestral locations for clones, whether they live in one, we have posterior distributions, blah, blah, blah. But we can infer this, the history in time and space of a cancer. So we are saying that in 2009, there was a first movement within the primary tumor. It started here, the cancer, because we have many locations from the primary tumor also. Then 1 year later, there was a metastasis via blood, okay, via the porta vein. Then there is movement and movement back towards the lymphatic nodes, okay, and then again several movements back through the lymph again. So the blood to go up, the lymph to go down. And this reminds something called self-sealing, like homing, some kind of homing that attracts cells to go back to the primary tumor. This is described. It has been proved in mouse and things like that. But we can disentangle with a probabilistic approach the history in time of space of a tumor using trees. And I'm going to end, sorry. So I hope that these are the only 4 messages I want to give to you. So bulk trees are not phylogenetic trees. You can keep using, you are going to keep using. We will see sample trees in nature, I'm sure. They are not phylogenies. It's up to you. We can adapt known methods that have been developed over decades in phylogenetics, statistical phylogenetics to study, and in fact, in our conditions, given our simulations, there's always biases in simulations, they seem to outperform competing methods in our simulations. The tree is not the end of the evolutionary analysis, okay? I showed you how we use the tree to infer movements in space, in time, which at the end of the day is biology, is cancer biology. So we learn about whether this guy goes through the lymph or through the blood. That might have some relevance, right? For this particular tumor, right? And perhaps most importantly, it gives us a quantitative understanding of things. It's not a description saying I have a tree and then I make an interpretation by eye. No, we estimate quantities, we estimate growth rate, we estimate age. These are numbers. We might be wrong, yes, but we try to take into account uncertainty in these estimates, okay? So there's a lot of people involved in all this, like colleagues and agencies funding this, and pathologists, of course, all these people. And of course, I want to thank Nuria and Contra for the opportunity of talking here. Thank you.
Questions?
I might have missed it, but does the model that you propose follow the infinite size assumption?
Say that again?
Does the phylogenetic inference method that you showed follow the infinite size assumption? So infinite size model?
No.
It doesn't?
No.
I missed it.
No, basically this is a Markov model where you consider the probability of, from the nodes of a given tree, you calculate the likelihood, the probability of observing a state in this node and a state in the other. And then you don't care about whether there were 1 change or 1 million changes between. You have the probability of the beginning state and the ending state. You assume a Markov process, which I think is reasonable for DNA. So, no, we don't need to assume many things, really. We estimate everything from the data without many assumptions.
So how robust is the method if you don't just sequence single cells, if you have doublets and things like that?
Yeah, I mean, doublets is just error, right? So I have slides for doublets, but because I wrote a simulator, what is here, okay? So basically, it's better than the competing methods. Of course, it's sensible when you have a lot of doublets. 20% put it here, no? 20% doublets is a little better only than other competing methods, but doublets can be a problem or not, depends on the technique you use. If you use FACS and then you inspect your wells, we don't have doublets in our lab. So that's when you do high throughput, things like that, you might need. But you can modelize the doublets error is artificial, no, mutations that you have there that are present in the population. You can take advantage of this. But so this is the answer. It affects all methods to a different extent, but we seem to do quite well. But it's just another error. The parameter that will absorb this is our amplification error. It will absorb these errors somehow. I don't know if that's answers you.
Any other questions? Well, I'm sure there will be plenty of time in the coffee break. Thank you, David.